Hi folks, welcome to chapter 12, and this is going to be about the endomembrane system. So as before, there are going to be several parts to some of these sections, so I've just broken the sections up into smaller pieces so they're not too long. First thing we're going to get into is a little bit about the endoplasmic reticulum. So this is the first part of the endoplasmic reticulum. So, as you probably know, the ER, the endoplasmic reticulum, is a continuous network of flattened sacs and tubules and vesicles that run throughout the cytoplasm of the eukaryotic cell. We call the sacs the cisternae, and then the space inside them where all the biochemistry occurs is called the lumen. And those are words you've probably heard before when you've been talking about things like the Golgi, and lumen has probably come up when you're talking about the chloroplasts. So we'll talk about rough and smooth. Now, when we just think about ER in general, the enzymes that are associated with the endoplasmic reticulum are often involved in the synthesis of proteins. So they were talking about rough endoplasmic reticulum. And so these would be proteins that are going to be incorporated into the plasma membrane, or they're going to be exported from the cell by secretion, such as the case of any protein hormones, things like that, small signaling molecules, things like adrenaline. They would have to be produced in the ER and secreted into the extracellular fluid. And the ER is also involved in lipid synthesis. So those are two very kind of broad categories of function we can think about. On the left here you can see some rough endoplasmic reticulum categorized or characterized, I guess, by multiple bound ribosomes involved in translation. And then on the right hand side, smooth endoplasmic reticulum. Now when you see these diagrams, bear in mind that sometimes the ER won't just look like these long thin ribbons like it does on the left. If we cut them in a different orientation, we may be actually looking down the middle of a tubule, for example. And so in this case here, this would be the lumen, whereas here this would be the lumen with the ribosomes on the outside. So a little bit by way of introductory material for the ER that you're probably a little bit familiar with. You're going to find both rough and smooth in most types of eukaryotic cells, but there is variation in the relative amounts of each type, and that's going to be related to the function of the cell. So here we're getting into how the structure of the cell and the components found in the cell relate to the function of the cell. So if we had a cell that's involved in secretion, then you're going to see a lot more rough ER, because there's going to be a lot more translation of protein occurring into the ER, so that protein can then be packaged and sent through the endomembrane system to the cell membrane, where it would be secreted. So the example here we've got on the left is a plasma cell. That's a cell you find in the immune system, and it secretes antibodies, and so that's a secreted protein, and so we're going to see a lot of rough ER there. Now, on the other hand, a cell producing a lot of steroid hormones, for example, a testicular cell producing a lot of testosterone, that's going to have more smooth ER, and smooth ER is more involved in lipid synthesis. Now remember that steroids are a form of lipid. So although there is some ER in the testicular cell that's rough, that does have some ribosomes associated with it, the smooth endoplasmic reticulum is going to be more abundant, and so again we see a relationship between structure and function there. So let's talk a little bit about the protein synthesis and folding business of the rough ER. The ribosomes that you find on the cytosolic face or the cytosolic side of the rough ER membrane, they're going to synthesize both membrane-bound proteins and soluble proteins. So if you think about any protein that has to be in the ER membrane itself, or in the Golgi membrane, or in the vesicular membrane, or in the cell membrane, that has to be translated on the surface of the rough endoplasmic reticulum and threaded and transferred so that the transmembrane domain spans the membrane of the ER. Now, proteins which are soluble would be translated directly into the lumen of the ER. So we have what's called co-translational insertion. That's when you have a membrane protein being slid into the membrane of the ER. As well as translation, the rough ER is the site of the initial addition of carbohydrates called glycosylation, so you're probably familiar with that. Folding of proteins, and the recognition and removal of misfolded proteins. We also see in here the assembly of multimeric proteins. So when we're going to have things like quaternary structure appearing, that can happen in the ER lumen. Inside the ER, there is an ER-associated degradation system which can try and refold, and if it can't refold and can't modify appropriately proteins that are found in the ER lumen, then they'll be exported to the cytoplasm for degradation, and that occurs via a group of proteins called the proteasomes. So what are we looking at here in terms of translation? Is what we've got is a ribosome up here with a piece of mRNA, and if you remember, the mRNA is going to be read 5' to 3', so that's a piece of mRNA being read 5 to 3, and that translation initiation has occurred in the cytoplasm. So to start with, all translation initiates in the cytoplasm, and the first little piece of protein or polypeptide that appears out of the ribosome contains a sequence of amino acids called a signal sequence, and that signal sequence can be recognized by this small protein here, which is shown in green, called the signal recognition particle. Now when the signal recognition particle recognizes this little short chain of amino acids at the start of the polypeptide, the N-terminus of the polypeptide, the SRP binds that, and the ribosome can interact with what's called an SRP receptor on the surface, on the cytosolic surface of the rough ER, and that docks the ribosome at the ER. So translation starts in the cytoplasm, and then it occurs, the rest of it occurs on the surface, and so there's a translocation event, and so ribosomes, remember, they're diffusing around in the cell, and if the SRP interacts with the SRP receptor, the ribosome is then docked onto the surface of the ER. So it's not that you have these ribosomes on the surface of the ER and the mRNA comes to the ribosome. You always start translation in the cytoplasm. So once the SRP receptor binds to the SRP, is what we then have is the signal peptide is threaded through this structure called the translocon, and then the remainder of the amino acid synthesis in the polypeptide begins, and then the signal recognition peptide is actually cut by a signal peptidase, which is on the luminal face of the ER and associated with the translocon. So once the rest of the polypeptide is threaded through and into the lumen of the ER, the signal peptide's cut off, and then there's folding and assembly of the protein into multimeric complexes, and then at that point, that's when we see the quality control of proteins that are found in the ER. If they're not folded and they can't be refolded correctly, then they tend to be translocated into the cytosol and degraded there, and we'll come back to that later. A little bit on smooth ER. Now, smooth ER, even though smooth ER is contiguous, it's continuous, it's all one network with rough ER, the functions differ when you compare smooth ER and rough ER. And so smooth ER is involved in processing and storing non-protein molecules within the cell, so there's a lot of lipid metabolism done there. There's a lot of drug detoxification that occurs in the smooth ER, and that often involves a process called hydroxylation. That's the addition of a hydroxyl group to generally a relatively insoluble drug compound, and that increases their solubility. That makes them easier to excrete from the body because they can be dissolved in fluids which are more easily secreted. And that hydroxylation is usually under the control of a large group of proteins called the cytochrome P450 family, and they're often called the monooxygenases. So those monooxygenases are involved in detoxification via hydroxylation. So that's the end of our first part on endoplasmic reticulum, going pretty quickly, kind of dense. I'll be back next time with some more on the endoplasmic reticulum.